Hi, welcome to this lecture on EnvAdapt70 animals. Now we've been discussing on this water balance, that how we can maintain or regulate, or then might be that we might change the osmotic pressure and how much we can tolerate a different amount of water in our body. And one thing that we must discuss yet is on the anhydrobiosis. So living without water. So how we can tolerate the water loss is that we have different ways. First of all, if we are having hyperosmotic body fluids, so then means that you have plenty of different compounds and therefore there is osmotic flow of water in your body. So you're limiting then the osmotic loss. Then the other way is that you can have minimal water excretion. So you have concentrated urine, you have concentrated feces, you are not losing water on that way. But we have to remember that this is not can be done if we are breathing, because as a terrestrial animals, in the breathing we are all the time losing water. And then, of course, if we are drinking water, eating, etc., but then in the aquatic environments, definitely in the sea, there's the problem that actually there's the salt accumulation. And then the third version is that if we are having higher metabolism, then we are producing metabolic water. But of course the problem can be that, do we have enough energy stores for this high metabolism? And one way is that, we can have also, if losing the water from the cells, then the problem can be that in the cells, all these structures, whether they are proteins or lipid bubbles, etc., there's always water around them. So they collapse if we are getting all the water from the cells. And therefore we need some support for the cell structures. The dehydration is actually linked to a life with a frozen body that we were discussing elsewhere in this course. So in amphibians that are partly frozen, when there's ice in their stomach, etc., they are losing plenty of water during this freezing period. And that causes drastic concentration differences. The urea is very high. Glucose level is extremely high. If there is accumulation of sodium, there is accumulation of chloride, and there is a clear difference in the osmolality. So therefore this problem is not only that you try to avoid the freezing, but actually you are trying to avoid to get dry. And this dehydration is affecting on different, so in the osmolality and composition of plasma, and it might be the problem that is limiting the tolerance for freezing. And the problem is that, when you have this frozen body, there is no mental activity by which you can change your behavior or start pumping ions away from your body, etc., because you don't have that energy. And the similar changes are also happening not only with the freezing body, but for example in the air-breathing fish. They are staying there in the mud for maximum can be 1 year, and they are releasing a mucus covering. So when you have a cover around you, then you are limiting the loss of water, but might be that it's not enough. But then we have animals that can survive anhydrobiosis, so living without water. So they're minimizing the loss of water to reduce the amount of intracellular water. And of course, it cannot be complete because we must always have water to support the structures, and in some invertebrates are very good in this. And in some cases, it's not only to help you to live through this dry season, but also against the pathogens. So in this picture, we have a rotifer that is surviving at least 3, at least 4, even 5 weeks without any in the very dry places. So there is an anhydrobiosis of this animal. And the good point is that, okay, the fungus is dead. So these fungus are killed much sooner than this rotifer. And therefore, practically after 2 months, these remaining population of rotifers, there's no fungus left. So that's a good example of why you do something to live through this anhydrobiosis. So live through this dry season is that okay, now you can escape from the pathogens, etc. And one example of anhydrobiosis is the water bear on the turkeys. And they have 4 pair of legs and they are practically everywhere. You can find them in the 5,000 meter deep in the Indian Ocean, very high in the Himalayas, etc., hot springs, and there's about 900 different species, and they are very small, maybe 1 millimeter long. And they still have a complete digestive tract and some kind of brains also. And they are not real arthropods, but there's a sister group, so very far related to bees and other bugs. And they can lose 95% of the water in the body. And that of course means that okay, now they are reducing also the water inside the cells. And they are tolerating practically everything as an inactive cyst. So in this cyst form, you can't see any more legs. Etc. They are completely inactive. And they survive in this cyst form where there's a surface layer is formed. And so there is a very minimal oxygen. Water is passing through this cyst because it's a very thin layer. And with that, we can survive amazing conditions. Even 150 degrees of Celsius, even minus 200 degrees of Celsius, even few minutes in almost in the absolute zero. Very high radiation. So 1,000 times more radiation than we would be dead. Even the vacuum of space. And therefore, they've been even trying to put these animals in space every once in a while to test that, okay, how they're working. And one way that these extremes can be done is by using a sugar called trehalose. So this trehalose is produced from the glycogen by the glycerol 3-phosphate. So you're using only the glycolysis pathway. So there is no need for ATP to produce this. And there is no need for oxygen to produce this. And it's initiated, when it's initiated, there is a reduce of glycogen concentration, of course. Because now you have activated this process not only to produce ATP with the glycolysis, but to have this trehalose. See, so these modified sugars. And they've been used quite much in the anhydrobiotic animals. In several animals, water bears, brine shrimps, these nematodes, the animals that are surviving with maybe 20% water left or whatever. They can use different sugars, in some cases a combination of sugar and protein. And over here we can see two different water bears that during the dehydration, this animal is increasing the trehalose very drastically and then putting the trehalose down during the rehydration. But the interesting part in this is this lower picture, where we have another water bear. There is no trehalose at all. So it's not the magic bullet. It can be found in some animals, very high concentration. Brine shrimp is a very good example. So maybe 18% of the dry weight. And it affects on two processes. First of all, it will bond with the macromolecules and cell structure to place the water. So it can support the cell structures. And then it can also be highly concentrated in the remaining water. So it's not anymore liquid water because it's having so huge amount of trehalose. So now it's more like a gel. So it's a solid liquid. And therefore this gel or solid liquid is not anymore losing so much water. Thank you.